So the vax is super safe and effective and all that stuff. John Campbell's got here's some concerning information about the vax that you didn't know. Get this. Did you know that there's 2 different batches? There was the batches they tested, then there's the batches they gave you. And they weren't formulated the same. Did you know that? Watch this.
So you're an academic, Josh, at Hebrew University, but basically you've got links with the States as well, though. And you've been studying some pretty interesting things. And the first one we wanted to talk about is the different processes that were used in the production of the Pfizer vaccine. Now, there seems to be process 1 and process 2, and this has got particular processes were used in the trials. Just give us a bit of background on that, Josh, if you don't mind, please.
Okay, yeah, sure. So the key point here is that in the clinical trial that Pfizer BioNTech ran on their COVID vaccine, they used vaccine doses that were made with one manufacturing process, what you refer to as process 1. But what was given to the public after the trial was not the same type of doses. Instead, they used a different manufacturing process, an upscale manufacturing process they call process 2, and that was what everybody received when they got vaccinated. And now the question is, well, what's the difference between these 2 processes? Does it matter? And it probably does matter a lot. And I can walk you through some of the evidence of why we think it matters. But just to give you sort of the general background, and I don't know all of the technical details of this. I know enough to kind of understand it, but we all have learned about So what do you need to do to make an mRNA vaccine? You need to generate mRNA. How do you do that? Well, mRNA is like a copy of a part of a strand of DNA. So you need DNA as sort of your backbone template that you can then create mRNA off of that DNA, okay? So where do you get that DNA from? Well, you can, with the process 1, they were basically using a PCR process to duplicate DNA, right? That's what they do with PCR. They want to check, do you have any of this remnant of DNA in your nose that we can say was, COVID, was SARS-CoV-2 or whatever, right? So they replicate it, right? That's the whole idea behind the PCR. And it's relatively clean to do it that way. And they were also using a very, very high quality system using these sort of magnetic beads to take out any impurities after that PCR process was used, again, to create the template where then they use that to generate the mRNA. In process 2, they use a different method to generate the DNA backbone, okay, where they basically have a DNA template that they put into this thing called a plasmid, which is part of a bacteria, okay? And so when the plasmids can duplicate, and then the bacteria also duplicates. And it turns out to be a far more cost-effective, scalable way. You can just have, instead of like having to put it in a PCR machine, you just have these big vats of, I don't know actually how big they are, but you have these vats of bacteria growing, and they can replicate very, very quickly. And then you take the plasmids and you take their DNA and they make it linear instead of like wrapped up, and then they use that to generate the mRNA. But after that point, what they need to do is they have the plasmid DNA and they have the bacteria still in that goo, and they need to clean that out and purify it. And that's where they've seemed to have done a terrible job. Oh. And I don't know if you've been following any of the revelations that are coming out, where more and more researchers around the world are replicating this finding that there are high levels of this plasmid DNA remnants in the vials of the Pfizer vaccine that have been tested. In addition to that, we know from Pfizer's own documents that they've submitted to various regulatory agencies that there's some residual lipopolysaccharide, otherwise known as endotoxin. This is the membrane. So what is the bacteria that they use for duplicating this? It's E. coli bacteria, and which is a type of bacteria that's known as gram-negative bacteria. And the membranes of gram-negative bacteria are highly inflammatory. They're called endotoxin for a reason, right? And so there are higher levels of this endotoxin in the process 2 vials. And again, this is based on what we know from Pfizer's own filings to the regulator.
Can I just see if I've got this right so far, Josh, if we just run this past you? So the process 1, they synthesize the DNA. I assume synthesize it in the right bases, in the right order. And then they multiply the amount of DNA with a PCR process. Now, I'm not a forensic specialist, but I think we probably do this in forensics, where there's a tiny amount of DNA found at a crime scene, for example. or in an archaeological specimen, which is a fascinating field. And they're able to multiply up the amount of DNA so they've got enough to sort of be able to recognize it. So they use this duplication process, and that PCR process, it's the same as is used in testing, where you duplicate the amount of RNA that's present. And of course, there's a debate about that and how much it should be duplicated and whether that can give rise to false positive results, of course, but leaving that aside. So here, the increased amount of DNA, they use that DNA to make the RNA. So the DNA is the code to make the RNA. So it's DNA makes RNA makes protein is the way it goes in biology. And that's trial process 1. But trial process 2 sounds a bit more like making beer to me. You put it all in some big vat. Instead of using yeast, though, you use bacterium. E. coli bacteria, we believe.
We know E. coli. We know a lot about their manufacturing method, and a lot of it came through the EMA data breach. Other writings about it. There's publications. We know. It's E. coli.
Mm-hmm. That's good. So the process 2, you kind of make a batch. Now, a plasmid is basically a way for transferring genetic material from one bacterium to another, as far as I understand it.
I think that's right. Yeah. Yeah.
So they're using this kind of way. The DNA is in the plasmid, and that means you've got bacterial components there as well, and we don't want DNA from the plasmid, and we don't want other things like bits of cell membrane left over.
Right.
I would imagine if you did have cell membrane in any injection, we're not saying the cell membrane in this, but if you did have gram-negative cell membrane in an injection, that would make you feel pretty rough for a period of time.
It could, and
it's toxic.
We'll talk about that later, how that might be linked to some of the adverse events that came up and have been,
So they're saying that this is. So he just said that there's. Correct me if I'm wrong. What I'm hearing is that there are 2 totally different processes for making the vaccine. The one that they tested, and then the one that they gave you.
Yeah,
it's not the same.
Yeah,
and the way we know this is from Pfizer's own data.
Yeah,
and now he's just said that this is being linked to adverse events.
Yeah, and it seems that they're doing this other way because it's more expedient. It's cheaper. Yeah. Even though it's going to have more adverse effect, their bottom line goes up, and it's just quicker, cheaper, and they can do it that way. I mean, that's what I think he's insinuating.
I think that's what he's saying.
Yeah.
But of course, the FDA says that the vaccine is safe and effective, Kurt. And it will keep you from being hospitalized, sick, or death. And it's fantastic. It's super safe. So let's listen to. He has a little bit more to say. Now he's talking about how it's connected to adverse events.
Written up in the mainstream academic press that we didn't see hardly at all in the trials. So we could talk about that later if you want.
Indeed. Now, have I got that summary roughly right, Josh?
Is that summary exactly right? And before we get past this, I want to make this point very clear: is that when you're making a biological medical product or biologic, the process is crucial. In fact, some people say the process is the product, because you're dealing with these very complex, biochemical interactions, large molecules. I'll give you an example. Have you ever heard of the Cutter incident?
No, please tell me.
The Cutter incident, and there was a book written by one of the high priests of vaccinology in the U.S., Paul Offit, on this topic. But what happened? The Cutter incident happened with the original polio vaccines in the 1950s. So Jonas Salk, created this polio vaccine. They ran a trial on about 2 million children. It was very successful. And so then they turned it over to a few different labs, and they said, okay, now we need to make a bunch of this stuff. But they didn't provide them with the precise directions on how to exactly replicate Salk's process.
Can you believe that? So I know Kurt brought this up on the show originally. That's how I first found out about this. The first batches of the polio vaccine, not good. So go ahead. So
what happened in the Cutter laboratory, they were making vaccines that had live polio virus. And when they injected them into kids, they got polio. There were over 200,000 kids who got short-lived polio, a few hundred who got permanent polio, and a few kids who actually died. Okay? This is not conspiracy theory. This is not anti-vax mythology. This is accepted history. And one of the key lessons that was learned from that is, when you're dealing with something complicated like a vaccine, a biological drug, you need to be very precise about the method. And once you change the method, you change the product. And you can't just assume that it's going to have the same effect on people. So you need to do another clinical trial on your new product. It's not the same product.
So that's where we'll end it. Mm-hmm. So I don't know how much I'm allowed to say, but I'm just going to show what John Campbell showed, because none of that's controversial. All that comes right from Pfizer's own data.
Mm-hmm.
And so nobody's is interpreting it. They're just reporting it.
Yeah.
That there's actually two different processes for making.
Yeah. And the process is the product was
is
the
what
thing
he
that
said.
really stuck with me too as well. But Jimmy, I want to say that polio situation, that was then. This is now, and today's vaccine is safe and effective.
That's
says
right.
the FDA and
That's
the CDC.
right.
So just remember that, pal.
I do remember that. Again, people are, I don't understand why people aren't upset that they've been lied to. That people still don't care. I had Cornel West on the show, couldn't care less. I mean, he's just, I'm not picking on him. He's representative of most people.
Yeah.
Just, and everything that went along with the COVID response, the fascism that went along with it, the unbelievable authoritarianism, the crushing of businesses, black-owned businesses, the firing of 70,000 healthcare workers. Yeah. Don't we care about our precious workers?
Yeah.
Lives shattered, and no one cares. No one cares. No one cares. They don't care that they were lied to on a scale their minds can't wrap around. But what they get angry at people that point out that they were lied to.
Yeah. Yeah. And they're still going along with it. And I shared you the pamphlet from the New York Times.
Yes.
They are still going to push it. I don't know if they're doubling down, tripling down, or quadrupling down at this point, but they're still going along with it. Regardless of all this information that's coming out, when they're saying, look, we're just providing you with Pfizer's documents themselves. This is not us making this up. This is them telling you.
I'll never get over it. I'll never get over it. I can now accept what has happened. What I can't accept is how people accept that it's happened and they don't care. That's the part that's blowing my mind. I can't. People who hate big pharma, people who hate corporate control of government, people who hate government, they all. Hey, come see us on tour. We're going to be in Tampa, Boca Raton, Orlando, Dallas, Houston, San Diego, Bloomington, Illinois, Indianapolis, and Levittown, New York. Wow, that's a lot of dates. See you there.